Excellent. Yeah, I see more people joining us right now, so that's great.
So you can click from there, and then there's like this pops up. So and there's like
All right now.
just look at it. Don't hit anything. Stream is my computer.
Sarah, are there people that you're knowing are going to join us that haven't joined us yet?
I know 12 people signed up and we have 12 right now. So I think that's
Oh.
everyone.
Okay. Great. I'm just going to check my email just in case someone is asking me for the link. Nope.
That's great. Dr. Audet, you're muted, but I could hear you through the wall. You're saying we're ready to go. All right. Cool. Well, it's a pleasure to be able to speak to you all. So my name's Dan Norton. For those of you who don't know me, I'm a professor here at Gordon, and I think the connection to the Neuroscience Club that I'll be drawing, or the topic I'll be talking about today, has to do with Alzheimer's disease and how Alzheimer's disease works, but also what we can, how we can exploit our knowledge of how it works to hopefully help some people and reduce some human suffering. So I'll share my screen and I'll start to show you guys a slideshow. Just some PowerPoint slides. Probably. Okay. So I will start by introducing Alzheimer's disease just in terms of confronting the enormous problem that it is. So I think you could argue it's the most devastating disease that we face as a society. It's the most common, the reason for that is, it's the most common form of dementia. And I just saw a chat. Sarah, did I miss that? I thought I already did introduce myself. But,
Oh, okay.
Yeah, sorry. My name's Dan Norton. I'm a professor of psychology here at Gordon. Sorry if I forgot to say that. Right. So talking about Alzheimer's disease. So it's really bad. I guess one more thing about myself. To introduce myself is that the reason I'm talking about this of all things is after I graduated from grad school, my first job was as a postdoctoral fellow researching Alzheimer's disease. So this is, some of my career has been devoted to this. So this is, I'll be talking about things, related to the people I used to work with and stuff like that. So, I was starting to say, it's the most common form of dementia, and dementia is really horrible. If you have the misfortune to know anyone who has dementia or had dementia and died with it, it's really one of the most awful things I think that can happen for human beings. Just the notion of essentially having your mind taken away from you while you're still breathing, and especially, it's the worse it gets, the less you're aware of that, which is kind of a blessing. But, for the people that are around you, just watching the person that you love become less themselves very slowly over a period of years. It's just really, really awful. Probably don't need to convince you of that, but just to remind you of that. So in terms of, if it were some rare thing that there's this awful disease, that would be one thing, but it's very, very common. So currently there are 5 million people alive with Alzheimer's disease right now, dementia caused by Alzheimer's disease. Let me just mention, there's oftentimes confusion. I think people are confused. In fact, once when I was like in middle school, my friend, good friend of mine, his grandmother got dementia, and he said, oh man, she got dementia and she got Alzheimer's disease. It was like everything at once. It was really bad, and he thought they were two separate things, but that's not quite right. Dementia is a condition where you're kind of losing your memory and stuff, or losing some of your cognitive faculties, and Alzheimer's disease is a disease that causes that condition. And there are other ways to get that condition, such as stroke is the main other way. But Alzheimer's dementia, there's 5 million people currently in America with that. So it's the 6th leading cause of death. And this is crazy, you're not gonna believe this, but 1 in 3 senior citizens dies with Alzheimer's disease or another form of dementia. I'm not sure how many would be with Alzheimer's in particular, but I would guess, at least half is probably from Alzheimer's disease of that 1 in 3 that dies with dementia. And so that means it kills more people than breast cancer and prostate cancer combined. So if you're not convinced yet, you have no heart, that this is a really big issue. But another thing to consider is the money side of it, which is that for one person, the average cost of care, if you get Alzheimer's disease, is over $350,000 in today's dollars. So that means, if you lump all those people together, that I was just talking about, the 5 million people that have it, that's about $300 billion a year in care that we have to pay to take care of people that have Alzheimer's disease. And that's projected to rise to $1 trillion in, I should have said 2050, not 2020, sorry. And that doesn't include the cost of unpaid caregiving. So, a lot of people will take care of their spouse who's sick, or take care of their mom who's sick, and they don't work because of that. They're full-time caregivers. Sorry, one sec, my tea is boiling. Okay. So that's not a cost that's involved there, but that's another big cost. If you know anyone who is a caregiver, or if you've ever had to do any caregiving for someone who has dementia, that's really quite the burden. So this is a big deal. Ronald Reagan, died with Alzheimer's disease. There's all sorts of, concern to the degree that he was affected by it when he was president. I was just reading about this as I was preparing for this talk, and I guess Bill O'Reilly wrote a book about Ronald Reagan, alleging that he was affected by Alzheimer's disease while he was in office. And some people deny that strongly, saying he did not have Alzheimer's disease while he was in office. He did, die of it 20 years later. In 2004, he died. I think he was in office till what, 88? But that's, yeah, he's a famous person with Alzheimer's disease. There's many other examples we could list. So to date, there's no cure for Alzheimer's disease, and there's actually no treatment at all that's effective. So it would be quite the bummer if my talk ended at this point. I painted a picture. We have this devastating disease for which there is no cure. Affects a ton of people. 1 in 3 of us who are experiencing this Zoom session together, 4 of us are going to probably get it, or at least some form of dementia before we die. And most likely it would be Alzheimer's disease. So I remember when I was in college myself, this was not my favorite, thing. I wasn't passionate about Alzheimer's disease or forms of dementia and stuff. I think when you're young, it's a very easy thing to say, that's a long ways away. I don't want to think about that. but yeah, I hope that this can introduce to you some of the reasons maybe it is worth thinking about, even though you guys are very far away from it yourselves. So the talk fortunately doesn't end here. So there's reason to have some hope, and especially for you all, if you guys are 20 years old on average, I think there's hope that there will be effective treatment for Alzheimer's disease by the time you're in the age where that's a big risk. So the treatments for Alzheimer's disease are being developed currently, and they're based on a hypothesis for how Alzheimer's disease works. And it's called the amyloid cascade hypothesis. So Alzheimer's disease was discovered by Dr. Alzheimer a long time ago. I think it was around 1900 or so that he discovered it. And the way he discovered is he had a patient who developed dementia. So, dementia had always been a thing in society. People knew that, people went senile. Was a word people would use back then. And Dr. Alzheimer had a patient who became senile, and this must have been around the time when certain ways to investigate brain tissue were being developed around 1900, and he was able to examine this woman's brain after she died with dementia. And what he found was that there were two characteristics of her brain. One was that these plaques were developing, like, the plaque on your teeth, like this smeary kind of stuff, was caked over stuff kind of throughout her brain. And that turned out to be, upon further analysis, the protein amyloid had kind of clumped together in parts and kind of smeared all over the brain in these plaques. And the other thing he noticed was that there were these little tangly things that were clumped in different parts of the brain, kind of all over the place as well. And they were made from a different protein called tau. So that was a big discovery by Dr. Alzheimer. That's part of the reason the disease is named after him. And he said, oh, this is a unique process where these amyloid plaques and tau tangles are arising. And I don't know if he decided to name it after himself or someone else did, but ever since then, this process where you get dementia and these plaques and tangles come, it's been called Alzheimer's disease since that day. So now the theory is that what's happening is that actually amyloid is beginning to accumulate in the brain, and that causes tau to accumulate in the brain. And tau, in turn, causes neurodegeneration. Cell death, basically, is what we mean by that. Somehow tau tangles are causing neurons to die in your brain. And then the neurons dying is what actually causes symptoms, what causes you to forget how to get home when you're at the mall. Something like that could be, one of the early symptoms of Alzheimer's disease. So that's the theory. That's the amyloid cascade hypothesis. Why don't I guess you can unmute yourselves if you need to, but why don't I pause there and just see how we're doing in terms of that theory and stuff about Alzheimer's disease. I really have no agenda other than to make this as useful for you guys as I can. So, and any questions so far, or anything you want me to go into further depth on? Okay, cool. Well, we'll proceed.
And I have a question.
Oh, yes, Brian. Dr. Rodden, please.
What about on the front of diagnosis?
Mm-hmm.
Does someone get clinically diagnosed with the condition?
Right. Well, technically, I believe it's still the case. This may change, as you'll see from some of the things I show you, but it's still the case you can't be diagnosed with Alzheimer's disease officially. When you have memory problems and it's looking like you're developing dementia, usually the first thing you'd be diagnosed with nowadays is mild cognitive impairment, which is like you're definitely not like you were, and we're worried that your memory's getting worse or parts of your cognition are you're on this kind of downward decline. Some people with mild cognitive impairment don't really follow that track and get worse and worse and worse. They just kind of stay mild. But a lot of people who are diagnosed with mild cognitive impairment would then be diagnosed with dementia after that. You know, years later, they might develop dementia after the mild cognitive impairment is noticed. And the official diagnosis in terms of Alzheimer's disease would be probable Alzheimer's disease, or dementia from probable Alzheimer's disease. And you could only know that it was Alzheimer's disease upon autopsy by looking at the plaques and the tangles in the brain after the person dies. There's no, you know, if you do an MRI or whatever, you can't see these plaques and tangles in the living brain. You'd have to wait till someone dies. As it turns out, we've recently developed the technology to image the plaques and the tangles. So I suspect it may be possible to diagnose someone with Alzheimer's disease in the not too distant future while they're still alive. But yeah, it's interesting, interesting area, diagnosis. Well, so this was the next thing I was going to show you. So this is the way that researchers view Alzheimer's disease now. Alzheimer's disease researchers view it, where, you know, cognitive functioning, okay, normal aging involves a slight decline of cognitive functioning. You're not quite as sharp when you're, you know, 70 as you were when you were 20, or even when you're 50, you're not quite as sharp as you were when you're 40, but this is a very small difference. And, you know, there's other things you get better at as you get older. But, you know, when you get really, really old, you know, you do get fuzzier and fuzzier with certain things. That's normal aging. When you're on the track, when you have Alzheimer's disease, that normal trajectory is accelerated so that your cognitive function declines at a faster rate. So this is here, you know, someone with mild cognitive impairment, their cognitive functioning has declined quite a bit. And then it's going to decline quite a bit more once they get dementia. And ultimately, you know, they will die from this because of the disease processes that are going on in their brain. The hope here is because of this stage right here called the preclinical stage. And this is the stage where you're on that path to Alzheimer's disease, but you haven't even gotten mild cognitive impairment yet. There's really maybe no noticeable difference about you in terms of your cognition when you're in this preclinical stage, but you're still sort of destined to be on this course, unfortunately, to develop Alzheimer's disease later on down the line. So for researchers, there's a lot of interest right now in trying to understand this preclinical stage better, because we feel like that might be the point where we can actually do something to help someone. Do you guys remember the beginning of the coronavirus, where everybody was talking about, you got to social distance to flatten the curve, right? Everybody wanted to, in terms of the spike of coronavirus, they wanted to flatten the spike so the hospitals didn't get overwhelmed and stuff. This is the same exact idea. In fact, I wonder if the coronavirus people stole it from Alzheimer's research, because we used to talk about flattening this curve, except in the opposite direction. What if we could slow this curve down at least, you know, make it less aggressive, or even make it the same slope as normal aging? Then that would be really, really good. So that's what the hope is right now. Once someone is at this point, they have dementia. I don't have too much hope that in anyone's lifetime right here who's listening to this Zoom talk, that there will be any treatment that we can do for someone like that. Because why do you have dementia? Well, go back to the amyloid cascade hypothesis, because a lot of neurodegeneration has already happened. Many neurons have died already, and I don't know of a way to bring, you know, those neurons back to life. Maybe, you know, there will be a way to do that someday, but that's much further off. It's much more promising to be able to intervene in the stages where amyloid is accumulating and tau is accumulating. If you can slow those things down or even stop those, then neurodegeneration will never happen, or at least it will happen much slower if you could slow down amyloid and tau accumulation. So that's really the hopeful part about Alzheimer's right now, is try to flatten this curve. Okay, does that make sense? Questions about that?
Yes, I have a question.
Yes.
How do you know if you're in the preclinical stage?
Ha, the rest of my talk.
Oh, okay, thank you.
What a great question. Yep. Very, very tricky to know. Yep. None of us could know, for example, for ourselves right now, but the hope is maybe we can develop a way to do that, to identify people that are in the preclinical stage.
So if my grandmother, she has Alzheimer's, is it genetic? Does that mean I'm going to have it as well?
Another great question. Glad you asked that. It is, well, it's not like Huntington's disease, or, other genetic disorders where that's like, if my dad had Huntington's disease, which fortunately he doesn't, there would be a 50% chance that I would get it because it's based on one genetic mutation. And if I get that particular gene from my dad instead of from my mom, I'm definitely getting Huntington's. Alzheimer's is not like that in almost all cases. We don't know why people get Alzheimer's disease actually still. There are some genes. There's one gene called ApoE, and it makes a protein or whatever that is in your brain, but there's different versions of that gene, and that affects the likelihood, like it'll, affect the risk from being, 1 in 10 to, 1 in 5 or something like that if you're 80 years old or something. So it affects your risk, but there's nothing that determines you're definitely going to get it. So there's some familial risk, but there's no familial determination. So for example, if your grandmother had it, it does not mean that one of your parents is going to get it. It does not mean that you're going to get it at all. In case to mention, there are things you can do that reduce the likelihood you're going to get Alzheimer's disease for yourself. So one of them is exercising. So you should definitely exercise throughout your life. And the other is being bilingual, which it might be too late for some of you, unless you really want to devote time and learn a second language. But I think it's more for people that are raised bilingual. That seems to have a protective effect against Alzheimer's disease. Mostly the exercising thing is the thing to remember for most of us, I think. All right. So this is needlessly complicated. This is just, I'm going to skip this. It's the same, saying the same thing as this prior thing. Amyloid leads to tau, leads to neurodegeneration, leads to symptoms. There's a couple nuances there, but I'm going to skip over that. So now this is Elizabeth? You asked, how do we know if someone's in the preclinical stage? And this is how we're beginning to know if someone is in the preclinical stage, kind of. This is several different graphs, as you can see. You see all these different colored little lines, and what's on the x-axis is what clinical disease stage you in. How far along in the Alzheimer's disease progression are you? So we're graphing, people. This is people with Alzheimer's disease. And the y-axis is just how much of whatever thing do you have. There's different things shown here. So a little is low and a lot is high. And where are you in the disease stage? So let's look at the brown line first. That's clinical function, the brown line. So the brown line is you don't have much disruption in clinical functioning. Sorry, I should have said abnormal is high on the y-axis. So it's normal for a while. Clinical functioning is normal. And then once you get to MCI, it starts to go up a little bit. So you have a little like clinical level dysfunction when you get MCI. It starts to be noticeable during the MCI period. And then it gets really, really bad when someone has dementia. It continues to get worse and worse. They can't take care of themselves basically, and stuff like that is what we mean by disrupted clinical function. So that's the last thing to change in this series of things that are shown here in this slide is clinical function. Cognition is the second to last thing to change. So here someone's in the preclinical stage, or they're totally normal for a while. In the preclinical stage, they're not showing any cognitive changes. And right around the beginning of MCI, they start to show some cognitive changes. Things that if you have a really sensitive test, you can see, okay, that person is not performing like they should. And that gets worse and worse during MCI, and it gets really bad during dementia, that someone will do really poorly on cognitive tests. Okay. So what we're looking for, if we want to identify someone in the preclinical stage, we want to find things that go up before we reach MCI as early as possible in the disease process. So let's see what goes up early. Well, the first thing here is called amyloid beta accumulation. So that would be, Dr. Alzheimer, that's what he found in the brain, was amyloid plaques. And you can see very early on in the preclinical stage, there's no years listed here, but this is early. This is starting to go up, okay? Amyloid plaques are beginning to develop in the brain many, many years before MCI begins. And if I had to put labels on this, it would be like more than 10 years before MCI as these plaques begin to develop. So that is a really good hope for trying to identify people in the preclinical stage. If we can somehow measure in the brain these plaques, we could predict who's going to get sick and who isn't from Alzheimer's. And at the very least, force them to exercise, maybe to slow down the thing. Or maybe we could have some pills or something we give them to try and break up the amyloid plaques, which have been developed, pills that do that. So that's the earliest thing to change. The next thing is synaptic dysfunction. So this would be measured like with fMRI would be one thing you might be familiar with here is people make the argument that that's measuring synaptic function, but basically, it's which parts of your brain are active, where's the blood flowing in your brain when you're doing particular tasks. So usually this sort of research would be like a memory task. You're learning new things, and your hippocampus is very active as you encode new information. So looking at the blood flow in your hippocampus for fMRI, that's something people are going to be a little abnormal on even well before MCI. So that's another promising area potentially to identify people in the preclinical stages with fMRI. And the last one I'll mention here. Oh, there's two more I should mention. One is tau. This is tau-mediated neural injury, but just you might as well forget the second half and just say it's tau. Tau begins to accumulate in the preclinical stage as well. So that's another way we could potentially identify people. And then finally, changes in brain structure. So the cortex tends to thin out in certain areas in people who are on the road to Alzheimer's, and that happens before any symptoms are noticed in the MCI stage. So these are all potentially useful pieces of information.
I have a question.
This is a complicated graph, so yeah, I was going to say questions on this.
What age do these plaques start to build up typically?
Right. Well, it really depends because Alzheimer's disease is very variable in when it onsets. But I would say, a good decade before MCI begins is when these plaques will begin to develop. Maybe 2 decades before, even could be that early. So if someone is going to start to get MCI when they're 72 years old, this would be noticeable when they're 52 potentially, or certainly by 62. Yeah.
And I have another question following up to that. So I know you do have done research with schizophrenia as well, and people with schizophrenia, they tend to have larger ventricles, so there was like more cell death. So I was wondering, are the buildup of these plaques also apparent in people with schizophrenia?
No, amyloid. Yeah, this has nothing to do with schizophrenia. It's a good question. With schizophrenia, it's less cell death than it is either the brain over pruning itself or just the brain developing differently to begin with. Yeah, but really good question and distinction there.
I had a question as well.
Yeah.
Does the amyloid, how does it build up in the brain? Does it come from food or is it?
Oh, wow, great question. Well, just trying to think of how ambitious I should be, because that's not really my area, but mostly I don't know. But I do know that it is something that your brain normally has to deal with, like amyloid proteins will be used in cells, and then they will kind of have to be thrown away in the trash of the brain, like your cerebral spinal fluid is supposed to like flush it out or something. And for some reason, it just gets, your brain, when you're on the path to Alzheimer's, is getting overloaded. You have too much of the amyloid proteins. Everybody has some amyloid proteins, floating around that have been discarded in their brain, but it gets just to be too much and they start to stick to each other and stuff. Your brain is supposed to break them down into little chunks, I think, and they're easy for, your cells to get rid of, but they stop being broken down as much in Alzheimer's and they start forming these plaques. So other people I used to work with at MGH would have been able to answer that question very well, but that's my level of sophistication when it comes to that. I don't think it's so much about food, though. That's another good question. All right, so preclinical diagnosis is really crucial to help people. And I'll just go back to this one for 1 sec. So can you guys see my cursor right now when I'm moving it around? Yeah. So there have been clinical trials already that have attempted to, people who have MCI, they have given them pills that break up amyloid in their brain. So someone comes into the office, they're kind of forgetful, but they definitely don't have dementia yet. They have mild cognitive impairment. And there have been clinical trials where you give someone a pill that successfully breaks up the amyloid plaques in their brain and reduces amyloid levels, which is brilliant. That's like, oh my gosh, you're going to win the Nobel Prize, you're going to cure Alzheimer's disease. But the problem is it did not cure Alzheimer's disease. It didn't do anything. It was not helpful at all clinically or cognitively. These people it didn't improve their memory or anything. So what people think is that it's really about this tau thing. So once you're at MCI, you've had amyloid plaques saturating your brain for like 2 decades at that point. And the tau cat is out of the bag at that point. And it's wreaking havoc in the brain. Amyloid somehow leads to tau. And if you start breaking up amyloid when someone has MCI, well, the tau is still already doing its thing. So apparently breaking up the amyloid doesn't really help that much. It's probably more about the tau. That's what people think. So that's why I say that preclinical diagnosis is really crucial. We really need to get at people before the tau begins to develop. That would be the ideal situation, at least. So how do you do that? How do you diagnose people preclinically? It's a really good question. So one would be to just take a ton of people who are like, say, 50 years and older and observe their brains and wait till some of them get Alzheimer's disease. And then we can kind of go back and be like, oh, what was different about their brain? And we could use those things as markers for Alzheimer's disease. And the graph I was showing you before is partly based on studies like that. That's a great thing to do, but it's a very expensive form of study. You have to have lots and lots of people in your study because only some of them are going to develop Alzheimer's disease. So it's very expensive. Another thing that would be good would be to study a few people whom you know in advance will get Alzheimer's disease. And that's great, but it's impossible because we don't know who's going to get Alzheimer's disease, right? And someone is saying something in the chat. Oh, we got a question. Is there a difference in how these factors will be researched for early onset? Oh my gosh, wow, another great segue. So Charlotte, I'm going to talk about early onset in just a second. Literally. So next slide. So here's one of the ways we hope to make progress is by studying people with a unique form of Alzheimer's disease called autosomal dominant Alzheimer's disease, which causes early onset Alzheimer's. So this is a rare case, which none of you have. Don't worry. It's the type of thing I might worry about if I were in your shoes. This is why I say that. It's a very rare mutation in a particular gene called presenilin-1, and it causes amyloid to accumulate in the brain at a very early age. There's something wrong with that protein and the way it cleaves when it's broken down, and it causes a ton of amyloid to accumulate very early. And people who have this mutation have a guaranteed onset of MCI at around age 45 and dementia at around age 49. So that's like 30 years earlier than it would be for regular Alzheimer's disease, right? So it's a terrible thing, but what it is a great scientific opportunity, because this you can do a blood test for. If someone has the presenilin-1 mutation, you can just take their blood when they're born, and you know that, that they're going to get Alzheimer's when they're about 45. And therefore, we can then do the thing I was mentioning a couple slides ago. We can study them really, really closely and see what's different about them that we might be able to use as a marker to identify preclinical Alzheimer's in the general population. So let me just introduce you real quick to a couple of the doctors I used to work with. This is Risa Sperling. She is in charge of a study called HABS, the Harvard Aging Brain Study. And this study is the first type of study I said, the really expensive one where we study a bunch of people and we just wait for some of them to get Alzheimer's disease. Oh, we're waiting on the results of the study you asked about, Emily, about whether we can prevent Alzheimer's in these people with AD. I'll let you know when the results come out, hopefully soon. I think I need to unshare for a sec because I probably need to reshare and make the sound work. Okay, I just want to introduce you to Risa. You guys are all at Gordon or you're at URI, right? So that means you're not that far from Boston. If this is interesting to you or anything related to this, Risa Sperling would be at the top of my list of people you should go try to get a research assistant job with after college. If you want to get your PhD in neuroscience or clinical psychology and then become a neuropsychologist or anything like that, Risa is really, really top notch. She's one of the best leaders and most intelligent scientists I've ever had the privilege of working with. Whoops, sorry.
I don't think the video is showing like full screen.
Sorry?
I don't think the video is showing like full screen. It was showing like a notes section and stuff like that.
Yeah, I probably shared the wrong screen, sorry. Yep, share. How's that, better?
My grandfather started showing symptoms at the time I was decided to apply to medical school. And unfortunately, he died when I was a neurology resident, so about 6 years after he was recognized to have symptoms of dementia. And I swore at that time that I was going to do something about it before I saw this in another family member. And unfortunately, last year, my dad was diagnosed with Alzheimer's dementia. He's a very smart guy, a professor, and so I knew things were changing for a long time, but I think he was able to compensate. And last year and this year in particular, he's really changed, gone downhill, and I'm seeing from a whole different level what it's like to deal with this as a family member, watching my mom having to deal with this, and I have to say it makes me sad, but it also makes me even more determined, I guess, to make sure that my children don't ever have to do that for me.
Okay. So there's more to that. You can check on alzheimers.org or just YouTube Risa Sperling if you want to hear her talk. She's a very excellent speaker. So she, as I said, she's in charge of that study, HABS, where she's studying hundreds and hundreds of people, waiting for some of them to get Alzheimer's, trying to characterize this preclinical stage so that we can hopefully, screen people and so forth in the future. The other scientist I'll introduce you to is Dr. Karosh. She was my direct boss when I used to do this postdoc at MGH. Essentially, Dr. Karosh used to work for Dr. Sperling, and then she got her own grant money and then hired me when I was done with grad school. So Risa Sperling is sort of like the grandmother, Yakel is the mother, and I was at one point the child of this scientific family that we're looking at.
For the past several years, I have been studying a Colombian family with autosomal dominant Alzheimer's disease. This is the world's largest family with one single mutation for autosomal dominant AD. So what we are doing now in my lab is trying to define biomarkers of some of the earliest brain changes associated with the risk to develop Alzheimer's disease. We have the unique opportunity to actually study individuals who are carriers of this genetic mutation, but they don't have any cognitive symptoms yet. So usually they develop symptoms around the age of 44, 45. It's like an early onset form of the disease. And the ones that are actually studying right now, they are between 25 and 44. So in that way, I actually getting a sense of what's going on before they start developing cognitive symptoms. What we have done in the past few years is trying to understand how amyloid and tau start accumulated in their brains of these individuals several years before they develop the symptoms. Our most recent work has actually proved that amyloid accumulation starts in the brains of these individuals about 20 years before they develop the disease, or in this case, the symptoms. And the latest work that we have actually showed that tau, that is the other protein that is affected in Alzheimer's disease, it started accumulated between 5 to 10 years before they develop the disease. One of the exciting things about the future is I was just recently awarded Narrow One to actually do a longitudinal biomarker study of these families. It's the first time ever that we're going to be able to follow out these individuals over time. And having the possibility to actually define or even validate biomarkers in these individuals actually offers like a lot of opportunities for clinical trials and the possibility of actually preventing Alzheimer's disease in the future. We can actually see what are the changes that are happening before a person declines, so maybe in the future we can actually do things to actually prevent that decline in other individuals.
All right. So let's see. Looks like there's a question in the chat. Oh yeah. No, it's expensive because the equipment. So Charlotte in the chat was asking whether why Risa Sperling's study is so expensive with hundreds of people. It's expensive because you're doing a lot of brain imaging with them. So you're doing fMRI, you're doing MRI, any kind of imaging of their brain you can think of, EEG, and you're doing PET imaging, which is using radioactive chemicals and stuff to image their brain, and all that. fMRI costs $500 an hour to do, and you usually do multiple hours with each subject, and you do it like every year. You're following these people every year, having them get a checkup, get these brain scans. So it's very, very expensive for those reasons. Paying the research subjects is not much of the budget of HABs, I don't think. They get a meager salary. I think they get like a coffee mug or something. Looking for my HABs coffee mug. I have one somewhere. All right. By the way, one quick plug for you folks who are interested in neuroscience is you should totally go get a brain scan somewhere. Like go to Brown or go to somewhere in Boston, like MGH or McLean Hospital or Boston University or something. There's all sorts of studies if you look on the internet where they will pay you, some money as a healthy control or something. If you're at least 18 years old, they're always looking for people to do studies. And it's great to just experience it, to go in there, and it's really scary. It's a loud machine and so forth, these banging sounds and stuff if you do an MRI. But it's really cool to be a part of it and get a real sense of what this stuff is like. So I recommend that you do that. When I was looking to get married back in the day, I was like 25 years old or something, working at McLean Hospital. as a research assistant, I didn't do brain imaging back then, but the way I paid for my wife's wedding ring is that I used to do these studies. And one time I ended up vomiting because they gave me some medication. They wanted to see how it would affect my brain, like a painkiller. But, you know, it was worth it because I was able to then afford an engagement ring. So just a funny story. Okay, so let me tell you a little bit about these people that Dr. Quiros and I studied in these people who have this terrible mutation. I'll try to go really quick because I know we're running out of time. There's this family from Medellin, Colombia, that a lot of them have this mutation. It's like they're like an extended family of like 5,000 people, and they tend to stick within the family a lot in terms of their marriages and stuff. It's like the cultural way. So basically they got this mutation from a conquistador a long time ago. And then it stayed with this family for hundreds of years since then. And as Dr. Quiros said, it causes Alzheimer's disease. So this is a picture that they had a 60 Minutes episode on this family. These are people from the family. And these are the people that we used to study in the lab. And, you know, they're just like normal Colombians, but they have this terrible thing, and they know, like it's part of their familial culture. They know that my mom had it and she died from it, or my mom's taking care of my grandmother right now, or Alzheimer's is just part of their life in this family. And it ultimately leads to death. And this guy was part of the study, but he's no longer living. I think he died at like 58 years old or something. But dementia usually begins at 50, and, you know, death would follow within the decade after that. So there's a bunch of brain imaging techniques that we would do in either HABs or in the study with these Colombian people, including MRI, fMRI, MRI looking at structure, fMRI looking at function, where the blood's flowing, what parts of the brain are active, EEG, and then PET imaging, as well as neuropsychological testing. So you don't necessarily have to do brain imaging to characterize the preclinical stage. You can maybe find some really good tests that you can give someone with pen and paper that really tests their function of their mind, and maybe you could get that to be sensitive to Alzheimer's. So each of these has its pros and cons. Let's look at PET imaging real quick in some more detail because we're running out of time. We'll just look at one. So this is two types of PET imaging. And the one on the left is called PIB. PIB stands for Pittsburgh Compound B. I think in Pittsburgh they discovered this molecule that they could essentially make a radioactive molecule that they can then inject in someone's arm, and that molecule will flow all around the person's body. It'll get into their bloodstream. It's a radioactive chemical, and they designed it so that it really likes amyloid. So if there's any amyloid in your body, this chemical, as it flows throughout your entire body, is going to congregate in places where there's amyloid. So if you look at this person's brain right here, this is an unimpaired normal person, someone who does not have the mutation, who's in their early 40s. And it looks like they have some amyloid in the middle of their head, but, you know, not too much. This is a carrier who's in their 20s, and you can see this person has more amyloid than the other person does, even though they're still in their 20s, 20 years ahead of when they're going to get sick. In their occipital cortex, their visual cortex, they have some, in a little in the parietal cortex. Whoops. If you look at 10 years down the line, this person has a ton of amyloid by the time they're in their 30s. All this amyloid all over the brain, basically. Now, while we're still on this slide, if you look at the tau is not accumulating in a healthy person, nor is it accumulating in the person in their 20s who has the mutation, nor is it accumulating very much in the person who's in their 30s with a mutation. You have to wait for tau until the late 30s for tau to start showing up. By the time you're in your early 40s, when you have MCI, tau is all over the place. So tau is kind of the thing that really starts to show up right before people get sick, in the few years before people get sick. Amyloid apparently isn't that bad for your brain, other than the fact that it catalyzes tau, because you can have a ton of amyloid in your brain for like 2 decades, and it doesn't affect your cognition at all. At least that's what people think right now. That could change, but that's the state of the art. So this is just some PET imaging. So PET imaging is great. We can look at these proteins directly in the living brain. It's amazing. But there are some problems with it. Number one is it's really expensive. Number two, it causes cancer because it's radioactive juice you're flowing through someone. And number three is that there's really only 4 labs in the world or so that you can do this right now. One is at MGH, one is at the Mayo Clinic in like somewhere in the Midwest, and there's one in Australia, and there's somewhere in Europe, I think. So this is, it's really cool, but it's really not a practical thing that's going to help, you know, the population right now in terms of diagnosing Alzheimer's disease in the general population. Ideally, what we'd like is to be able to, you know, you get a colonoscopy, right? When you're in your 50s, as soon as you go to your checkup after you turn 50, your doctor's like, hey, got some news for you. It's time for your colonoscopy. And everyone's like, oh, I don't want to have a colonoscopy. But like, it's a screening thing to see if you have colon cancer. It's worth it. Everybody does it. And the goal here would be, what if we could do that with something like this PET imaging? What if when you turn 60, you get a PET imaging thing and we can see if you're in the preclinical stage of Alzheimer's disease. And if you are, we're going to try, you know, one of these medications that breaks up amyloid or something like that. So that's the basic idea of this stuff. I don't think I have time to really talk about anything else. I was going to say, what if we could use something else as a proxy for like tau building up? And I did this study on visual short-term memory. We were looking to see if performance on that test was correlated with tau. The long and the short of it was the correlations weren't that strong, so it's not that great of a proxy. So that's pretty much it. This will be my final slide, and then we can chat. There's a lot more work to be done on this, and you guys should get on it. You are going to be a generation that is, when you're in graduate school, you're going to be involved in some very exciting stuff if you decide to go into Alzheimer's research. So I will stop there and see if there's anything else you want to talk about in the rest of the time we have. Hi.
So you said that it starts building up when in just like normal Alzheimer's?
I mean, you know, 20 years, 15 to 20 years before MCI. And MCI can happen at many different ages. Some people think that if you were to live forever, you would eventually get Alzheimer's disease. Like, it's inevitable that you're going to get it at some point. If you live to 150, I'm sorry, you're definitely going to get it. That's not proven, but some people think that. I'm not sure if that's right or not. But you know, MCI can vary a lot, and Alzheimer's can vary a lot, and when it does come. But you have amyloid in your brain. If you do have MCI and Alzheimer's, MCI and dementia from Alzheimer's, that means you've had amyloid in your brain for at least a decade, like a lot of amyloid. Does that make sense, Elizabeth?
That's interesting. Thank you.
Yep.
Okay, I also had another question. Is there like a specific functions that start to go before others in like most patients?
Yep, definitely. I mean, memory being a key one that starts to go first. There's a lot of interest right now in that in terms of what are kind of the first subjective symptoms that you notice starting to go awry. And it's really tough because there's a lot of cognitive changes that are subtle shifts that occur in normal aging. So like forgetting where you put the car keys, or like having trouble finding the word that you want. These are cognitive things you notice, especially as you get older, that aren't very predictive of Alzheimer's disease. They happen more probably if you are on the path to Alzheimer's disease, but they're not something that would distinguish someone who's aging normally from someone who's on the path to Alzheimer's. I think the one that I remember from a study I read on this was that tends to be a little bit more common is that like if you forget how to get home from somewhere that should be familiar to you, like going, you know, to the grocery store or something, that would be, you know, kind of concerning as a first potential symptom. But so that navigational type of a memory, if that's really getting messed up, that would be an example of potentially an early sign.
With my grandma, I noticed it was kind of like short-term memory first. Like she couldn't remember things that had recently happened, but she could remember things that happened in her childhood.
Oh yeah. Yeah, I've heard people say that too. That's another good distinction is oftentimes those old memories, are really in there and they stick around, which is nice, to have something. But
Yeah, to add on to that, I know my grandma, she used to kind of like, it seemed like she was like going back in ages almost. So like she was like 70 or 80 and she would just kind of go back slowly. And when I was around, she would just kind of like recite her like child songs that she used to know. Like grandma was from Sweden. So she used to do Swedish prayers with me and like she knew those really well, but short-term things. She's like, oh, you've grown a lot, but it's just like, she just remembers me as tiny.
yeah.
But also another thing, I know my grandma who passed away this year, actually, her whole family, like her brothers and sisters, 7 out of the 8 of them had Alzheimer's.
Oh, wow.
Yeah, I've always found like the genetic thing weird, but like none of her like children have it yet. So it's like interesting to see.
Good.
Yeah.
Yeah. Oh, that's tough for your family to have to deal with that much.
Our time is winding down, and I wanted to take just a minute to thank Dr. Norton for putting together the presentation and spending an hour with us this afternoon, and this way supporting kind of neuroscience instruction within 2 clubs in 2 different states. So that's one nice advantage of Zoom, that we can connect people from different places. And so Dr. Norton, thank you for your time. Really appreciate everything that you've done in this particular area and your willingness to share with us everything. So again, thank you very much.
Thanks.
Thanks everyone else for joining the Neuroscience Club, really, and this particular talk that we gave. So thank you. Thanks.
Yep. Good.
Thank you, Dr. Norton.
Yep.
Yeah, thanks.